Today we're going to talk about medical school histology basics, cartilage and bone. We'll talk about the cells of both cartilage and bone. We'll talk about the conversion of the skeleton from cartilage to bone, growth of long bones, and we'll talk about the different characteristics of cells, the cellular characteristics of cells that make possible for the production of bone and the overall function of cartilage and bone. Thank you. Medical school histology basics, cartilage and bone. There's a host of cells that we have to be able to identify. There's chondroblasts, chondrocytes, osteoblasts, osteocytes, osteoclasts is another one. So there's a host of different cells that we have to do. Spongy bone or compact bone, cartilage, perichondrium, muscle. There's various things that we'll to talk about today. But what is the function of cartilage and bone? In terms of evolutionary and embryology model of bones, here you can see that before we had bones, we have cartilage as our tissue support. Also, cartilage slides across one another very easily like that, and it's somewhat a little bit compressible, and so it prevents bone from touching bone at the articular surfaces and somewhat of a cushion. Bone now supports land animals. Here we can see it also supports whales too, but here you can see this animal would not be possible to stand up without bones. It also protects its vital organs, liver, lungs, and it even protects the brain. Here we see the human with a skull protecting the brain, and it's in the bone marrow that we have hematopoiesis. So here you see bone marrow. Hematopoiesis is where blood cells are made. So cartilage is a structural support, but also in the adult you see it as a slipping slimy so that it can allow a movement in joints, and then bone is a major supporting structure of the body. Now remember, when we talked about connective tissue, we had some pluripotent cells in connective tissue that can produce blood cells, megakaryocytes that produce platelets. They can produce a host of other cells, fibroblasts, even endothelial cells, mesothelial cells, but also it produces osteoclasts, which is from the monocyte macrophage series that eats up bone, and it has osteoblasts, which lays down bone, and whenever an osteoblast gets trapped in this little space, it becomes an osteocyte. And in cartilage, we have a chondroblast, which get trapped in this lacunae or space, and it becomes a chondrocyte. So there's 5 different cells here of the connective tissue that we can see. Now if we start with cartilage, cartilage is composed of 3 different types. Hyaline cartilage is what you see at the end of a chicken bone, the white, glassy look interior. And that's what at the articular surfaces where one bone is touching another one at the joint. And it's not touching because of the cartilage in between. And then we have elastic cartilage, and we have that in the ear. And the function of elastic cartilage is return to the original shape. You can fold up your ear and it will pop right back because of elastic cartilage. And then we have fibrocartilage. Fibrocartilage is interesting because it connects tendons and ligaments to bones, and to where we can see normally this would be like a ligament with all the connective tissue that we see here, the type 1 collagen, but located right in there are the chondrocytes, as you can see. So here's a chondrocyte, which is located in this lacunae, and lacunae is equal space. So that's the space where the cell has laid down the extracellular matrix around it. So if we take a look at a joint, this is a joint of fetal elbow, and we can see the capsule around the cartilage, the perichondrium, and then we can see there's a host of cells, there's blue cells here, there's hyaline cartilage, and right at the growth plate, right in through there, the cartilage is changing to bone, is transformed to bone, and we'll talk about that. So here we can see that we have the hyaline cartilage, and this is going to be the hyaline cartilage are the articular surfaces, and you see the synovial membrane that goes around it, so it allows this bone to move in relation to this bone. And we have dense irregular connective tissue in the periosteum or the perichondrium, and there's a bone matrix. Now in terms of cartilage changing to bone at the growth plate that we see here, and actually even whenever you go from a cartilage model to a bone model, you have an area of reserve cartilage, and then we have proliferation where you see them all lined up there, and then you have hypertrophy where the increase in size, increased number here, increased size, and then it becomes calcified. When it does, the extracellular matrix is no longer diffusible. Nutrients can diffuse through cartilage, but it cannot go through bone because of the calcification. And that kills these cells here, and the osteoclasts, macrophage-like cell, moves in and eats that up, and it's replaced by bone. And we'll talk about that later on at the epiphyseal plate. Here we see hyaline cartilage. Here it is here, perichondrium on this side, and we can see characteristic of hyaline cartilage is this isogenic group. So a group, they have interstitial growth, so one cell divides to produce other cells, and a whole group of cells is developed from a single cell, and that makes an isogenic group that we have. This is epithelium on the surface of the trachea that we see right in through there, the respiratory epithelium, and then we have dense irregular connective tissue here on either side of the perichondrium. If we look at the ear, this is the outer ear, and we can see epithelium here, there's epithelium there, so we've got skin on both sides of the ear. But if you look at the elastic cartilage, it looks like hyaline cartilage. Isogenic groups are not as big, but you have elastic fibers in between, and that's what we see, the elastic fibers for the elastic collagen. And here we can see this is a pin of the ear of a cat, and you can see blue here is the elastic fibers that you can see there. So these are the chondrocytes that are trapped in the lacunae or space, and this is a chondroblast. So right at the edge of the perichondrium and the cartilage is where you have the chondroblasts, which grow by appositional growth. These guys differentiate into cartilage cells. But of course you have dense irregular connective tissue in the capsule itself. Now fibrocartilage is different in that it's between two different types, between a tendon, a ligament, and bone. And so here we can see the fibroblasts of the tendon or ligament, and these are big bundles of collagen, type 1 collagen that you see in through there. But right at the transition between this and bone, this is bone down here, you have these chondrocytes that are located in here, and they like differentiated from some of these fibroblastic-like cells, and they actually produce type 2 collagen, which you see a little bit darker right in through there, type 1 there, type 2 here. And so these are our chondrocytes trapped in their lacunae and the fibrocartilage. And there's no perichondrium. There's no perichondrium around it because they're already embedded in dense irregular connective tissue of the tendon or the ligament. So if we look at in the fetal elbow again, we look right here at the attachment, we can see lots of bundles of collagen in through there, and then we can see these little cells all stacked up like this, each one of them in their little lacunae, and those are the chondrocytes of the fibrocartilage. You can see them here, there, all in through here, and this is type 1 collagen. That would be part of the tendon or ligament. And also you can see skeletal muscle. So this is skeletal muscle. That is where the muscle is going to be attached to the tendon eventually, so that skeletal muscle would regulate location of the bone. If we see this in the cat, we can see our model again. So these would be like a tendon in through there, and you can see the fibroblast here, the fibroblast there, and then you can see the chondrocytes in the fibrocartilage. There's some right here, and you can see them in their lacunae right in through there. So you can see, and that's located right in this location right there's bone, and this cartilage attachment is right in through there where the bone is attaching to the tendon. So we can see the articular surfaces, that is where one bone touches another bone, and you can see there's a capsule around. And hyaline cartilage has a perichondrium except at the articular surfaces, and the articular surfaces of perichondrium will be worn off. That we can see through there. Articular is where one touches the other. Here we see a tibia, a fetal tibia, and you can see in this region in through there where the cartilage is very active, surrounded by the perichondrium, and there's a lot of activity here where you have the transformation of the fibroblast-like cells to the cartilage cell, and this is appositional growth, namely growth from the side. Again, we can see that this is a sulfonated gags. That's what makes it blue because of the negative charges that's on there. So this is cartilage, and these would be chondrocytes located in the lacunae, and this would be perichondrium, so this would be fibroblast in through there, and right in between there you have the chondroblasts. These are the cells that are transforming from the fibroblast-like cells that are located in the perichondrium, giving rise to the cartilage cells, the chondrocytes, when they get trapped in the lacunae, and that's appositional growth as it gets wider. So we can see that at electron microscopic level. There's fibroblasts in through there. There's a chondrocyte trapped in this lacunae. It produces type 2 collagen. However, in between there is type 1 collagen. And you can see the type 1 collagen right in through there with the periodicity that you have. So appositional growth is in this area, and the perichondrium is in this area. So these cells be transforming. Maybe this is a chondrocyte, a chondroblast, which will give rise to a chondrocyte. Now, one thing is characteristic about cartilage that you don't have in bone, and that is you have interstitial growth. Interstitial growth is where one cell divides to give another one. You can see here where the membrane has separated these two cells. So one cell gives rise to the other one, and you can have a bunch of these, and that's what happens when at the growth plate, when the cartilage is expanded for the length of bones, is that one cell divides, produce other cells, they line up, hypertrophy, die, and then that way the bone has increased in length. So here we can see in the embryo that we were cartilage. Our bone, our skeleton was cartilage at one time, and then it gets replaced, and then you have the epiphyseal plate where you grow. Before puberty, you start to grow, and the bone cannot grow from within. The cartilage has to grow, and then it's replaced by. And here we can see compact bone, which has these Haversian systems. So this is the osteon, and then these osteocytes that are located in through there. And you have lamellae, circumferential lamellae. So this is the end osteum, periosteum that we have with the bone. So here's the cartilage at the end, and then you have primary spongiosa. It still have a cartilage core. You can see the blue core in there, but this is cartilage and this is bone with a little bit of cartilage in the very. This would be compact bone on either side, and it would be the perichondrium and the periosteum is the capsule. So we look again at, you can see the densely regular connective tissue of the periosteum and the perichondrium that we see the hyaline cartilage. We can see this is also where you have fibrocartilage as well. If we look at the finger at the growth plate, again we got the different zones: zone of reserve, proliferation, hypertrophy, calcification. So that's a repeating cycle of all the various different bones that we have at the growth plate, which would be right in through there. So cartilage, primary spongiosum still has cartilage, and so are the different cells. The one cells on the side of these here are the osteoblasts, and the osteoblasts become osteocytes when they get trapped in their lacunae. Osteoid is bone. We can see all the pink here that we see here, compact bone. This is spongy bone in through here. The other cell is the osteoclast. Osteoclast is a multinucleated cell that we see here. We can see it there, more than one nucleus, and that's the one that breaks down bone. And so this one breaks down bone, and osteoblast lays down bone. When the osteoid goes all the way around it, it's trapped in its lacunae, and it becomes an osteocyte. Osteoblasts give rise to osteocytes. So here we see cartilage. All this is cartilage in through here. These are chondrocytes. These are the osteoclasts. Yeah, you can see one there. You can only see a nucleus, one nucleus there. And these guys who are trapped in the lacunae are the osteocytes, and this is compact bone, and this would be the periosteum that we see. A similar type thing there. Cells on the surface are laying down new bone, or it could be osteoclast eating up bone. There's the osteoclast that we see right there, osteocytes, and here we see the trabeculae. The projection that we see there are trabeculae, and we can see that this one still has primary spongiosum because it still has a cartilage core. Here we see electron microscopic view of the raphe reticulum, which is indicative of protein production, and bone is protein. Type 1 collagen is a main component of bone, and then you also have the hydroxyapatite crystals, which is what crystallizes bone that makes it hard. Secretory vesicles that we see, they're secreting the type 1 collagen, and you have a large Golgi apparatus which puts a sulfur on the proteins that are produced in the raphe reticulum to make collagen. So raphe reticulum, Golgi apparatus, secretory vesicles, nucleus, all these are osteoblasts. And here we look at toluidine blue. This is bone in through here, and this is cartilage. Don't worry about the different coloration, it doesn't matter. It was a staining problem. Here we see osteoclasts, which eat up bone. Osteoblasts, which are laying down, and this is a secondary spongiosum, doesn't have cartilage core in it. Osteoid is bone. Lacunae is the spaces that we can see there. Osteoclast, multinucleated cell, and these be osteoblasts laying down down more bone. Now, if you look at compact bone, there's several things. You got concentric lamellae, you got an inner circumferential and outer circumferential lamellae, and that was developed from appositional growth. And appositional growth is from the enosteum as well as the periosteum. So as a cell develops, it makes these layers here. In addition to that, you have the Haversian system. So this is a Haversian system here, and this is a Haversian canal. And the nutrients come through this canal, and it comes from the Volkmann's canal, from the outside through there, feeding the Haversian canal, which is where the nutrients go. And you can see this is a series of these Haversian systems, and they're long columns, and they run down the length of bone. They don't just run at random, they run down the length of bone. And what it is osteoclasts have gotten together to make a hole, and then the osteoblast fills in the hole. So here we can see one of these, which is just about a quarter of one, and you can see Haversian canal, cement, lacunae is the spaces, and lamellae are the projections between one cell, where one cell touches another cell and it gets nutrients from the gap junction. So from the Haversian canal comes blood vessels, and nutrients go through from cell to cell to cell by these lamina, by these caveoli. So these projections in through your caveola, these are different layers. One's laid down, another one's laid down. It lays down from the outside in, just the opposite of a tree for this Haversian system. Here we can see the Haversian system in ground bone. This is the Haversian system here. Here's a canal. These guys here are the osteocytes trapped in the lacunae. Actually, what you see is a space, is a lacunae that we see, and the canaliculi projecting from one cell to another, providing nutrients from the Haversian canal out to the various cells. We can see that again here. You can see the canaliculi, the lacunae that you can see, and inside there would be the different osteocytes. And so here you can see Haversian system. There's another one there, and as bone is remodeled, it eats up some new ones and some old ones. Here again we can see this is this inner circumferential lamellae that you see there, and then we can see another Haversian system in through there with a Haversian canal. So inside compact bone would be spongy bone. So this stuff right here would be spongy bone. So another Haversian system here and here where bone is remodeled. So this is a compact bone, this is a Haversian system with osteon, Haversian canal, lacunae, osteocytes that are located in through there, and the little spider legs in through here are the canaliculi. Here we can see unstained bone, so these would be the Haversian canals. Haversian system will be around that. Volkmann's canal is what attaches nutrients from the outside to the Haversian canal. Again, Haversian canals, and we can see osteocytes in through there in the Volkmann's canal. On this slide, we can see the osteoblast on the sides, which are laying down new bone, osteoid, which is new bone, a lacunae, osteocytes are trapped in a lacunae, and osteoclasts. Here's the osteoclast. This one and that one are osteoclasts, osteoblast right there. But here we see the cartilage at the epiphyseal plate. So you have bone up there, bone here, and cartilage in between there, which is growing by interstitial growth to make bone longer. Bone gets wider by appositional growth, but it's interstitial growth that it grows by the length. Length is interstitial growth for the cartilage because bone can't do it. Cartilage does it, and then it's transferred made to bone. But the width goes by apposition. And so here you can see the different zones of the cartilage, as in the reserve, proliferation, hypertrophy, and calcification. See those again and again. Here we see primary spongiosa. The primary spongiosa has cartilage in its core. The secondary spongiosa has osteoid in its core that we can see right there. So all these different regions, you have proliferation by interstitial growth of cartilage. Then you have the alignment as the cells are proliferated. Then they hypertrophy, then they calcify, then they're replaced. And that's just a cycle that goes over again at the epiphyseal plate. And the transfer of your cartilage skeleton to your bones. Here again, we can see the same type of thing with the different zones that we can see, the same different zones that we can see there. And the function of cartilage and bone, as we see in terms of summary, is in embryo, we had a cartilage skeleton. Some sharks only have a cartilage skeleton. And then for you to walk on land with all that weight there, you definitely have to have some bones to keep your shape. Also, you can see the cells of the Haversian system and the canaliculi, which allows nutrients to pass from one cell to another one, and of course the different zones of cartilage growth. A couple questions that may be of interest. Which are true about endochondral bone growth? Endochondral bone growth is where bone is used cartilage model, and then bone develops from that. And one answer is growth in width occurs by appositional growth. This is true. Okay, the growth in length occurs at the epiphyseal plate by interstitial growth of cartilage followed by replacement bone. True. So bone can grow by appositional growth. It cannot grow by interstitial growth. Okay, and so secondary spongiosa has an osteoid core. True. So the answer is E. Cartilage differs from bone in diffusibility of extracellular matrix. That's true. Nutrients can diffuse through cartilage but not bone. The presence of isogenic groups. True, because isogenic groups are produced by interstitial growth, and cartilage has it but bone does not. Ability of appositional growth. False. Both have appositional growth, and so the answer is D, A, and B. You ever see Big Bend? This is actually, these are mountains of Mexico at the distance that we see over there. So that's the end of the medical school histology basics, cartilage and bone. If this information was useful to you, please share it with your friends and your colleagues, and maybe consider subscribing to the VIBS Histology YouTube site. Thank you.